In this video, we'll look at the antibody and nanobody structure prediction data functions in ChemCharts. The antibody structure prediction data function can be used to predict antibody structures from just their sequences. In this table, we have 10 antibody sequences. The heavy chain and the light chain sequences have been concatenated into a single sequence with the heavy chain first. This concatenated sequence will be the input for the data function. From the data function settings dialog, choose the table containing the input sequences, the column containing the concatenated heavy and light chain sequences, and an ID column. Two models are available, A-Body Builder version 2 and A-Body Builder version 3. Click the checkbox next to the models you want to use. A-Body Builder version 2 uses 4 independently trained models. Choose whether to save all 4 predictions, or only the best predicted structure, or that structure closest to the mean of all 4 predictions. Predictions will be labeled by the model that generated them, and for A-Body Builder version 2, they're ranking among the 4 predictions based on the mean. The data function can be applied to all rows or a subset of rows based on marking and filtering. When the data function is finished, a new table is created containing the predicted structures. Click on the Configure Visuals icon to visualize the table in the Biomolecule Viewer. The new table has the following columns: the original ID of the sequence, the name of the model used for prediction, the submodel number and rank if it's available, compressed PDB structures which have already been aligned to the first structure in the table, a gene bank column containing the concatenated heavy and light chain sequences that are present in the predicted structure, and also sequences have been numbered using IMGT numbering, the original sequence provided as input, and the heavy and the light chain sequences present in the predicted structure. Clicking on a row in the table will display the structure in that row. Structures are displayed showing all heavy atoms and a cartoon of the antibody backbone. For A-Body Builder version 2, the cartoon is colored from white to orange based on the variability and location of that residue across the 4 models. For this structure, most of the predicted residue locations are consistent across the 4 models, indicated by lighter colors, with higher variability areas in the C-terminus of the light chain and the CDR-H3, indicated with darker colors. A-Body Builder version 3 uses a single model, so there is no variation in colors. Multiple structures can be visualized simultaneously by selecting multiple rows. The bottom pane of the Biomolecule Viewer shows the sequences of the displayed structures and their annotations. Clicking on a residue in the sequence will focus the structure on that residue and highlight it. The predicted structures can then be used with other antibody-related data functions. Structures for nanobodies can also be predicted from their heavy chain sequences. In the Settings dialog, specify the table and column containing the sequences and the ID column. Nanobody Builder version 2 is used for structure prediction. Like A-Body Builder version 2, 4 predictions from independent models are made and the best of all 4 predictions can be saved. The data function can be applied to all rows or a subset of rows based on marking and filtering. Once the data function is finished, a new table is created. Click on the Configure Visuals icon to visualize the table. Like antibody structure prediction, the visualization uses the Biomolecule Viewer and a table containing the predicted structures. The new table has the following columns: the original ID of the sequence, the name of the model used for prediction, the submodel number and rank if available for that model, compressed PDB structures which have already been aligned to the first structure in the table, a gene bank column containing the heavy chain sequence that is present in the predicted structure. Sequences have also been numbered using IMGT numbering, and the original heavy chain sequence provided as input. The visualization works the same as for antibody structure prediction. Selecting rows will display 1 or more structures. Backbone cartoons are colored by deviation from the mean of the 4 predictions for that structure. Sequences are displayed at the bottom and are responsive to clicking on residues. Thank you for watching, and check out our other data function tutorial videos.